ever imagined a world where science has the power to cure blindness? Welcome to the realm where science fiction morphs into reality, and the impossible is made possible. Today we're delving into the thrilling world of genetics, where a revolutionary tool called CRISPR is rewriting the rules of life. Picture a world where we can modify the genetic code of living beings, a world where we can rectify the mistakes that result in diseases and disorders. This is precisely what gene editing allows us to do. At its core, gene editing is akin to using molecular scissors to cut and paste DNA, the basic building blocks of life. This scientific revolution is driven by a tool known as CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. Quite a mouthful, isn't it? But fear not, it's not as intricate as it sounds. In essence, CRISPR is a defense mechanism found in bacteria that scientists have employed to modify genes. It operates by directing a protein, named Cas9, to a specific spot in the DNA sequence. Once there, Cas9 acts as the scissors, severing the DNA at that exact location. This enables scientists to remove, add or alter genetic material, effectively rewriting the genetic code. Now, let's bring this back to our initial question. Can science cure blindness? The answer, it seems, is edging closer to a resounding yes. Recently, a groundbreaking clinical trial led by Massachusetts Eye and Ear used CRISPR gene editing to treat 14 patients with inherited blindness. This blindness was caused by mutations in a gene known as CEP290. The trial, aptly named Brilliance, showed that the treatment was not only safe but also resulted in measurable vision improvements in 11 out of the 14 participants. This study, published in the New England Journal of Medicine, marks a significant advancement in gene therapy for ocular diseases. In the Brilliance trial, the 14 participants ranged from children as young as 8 to adults in their 40s, showcasing the wide reach of this inherited blindness. All of them had a history of poor vision from an early age, and all were ready to embrace the promise of gene editing. The procedures followed during the trial were meticulous. The CRISPR treatment was injected directly into the patient's eyes, targeting the faulty CEP290 gene. This procedure was performed under local anesthesia, and all safety protocols were strictly adhered to. The safety of the participants was paramount. Regular follow-ups were conducted to ensure that there were no adverse reactions to the treatment. These included thorough vision examinations and tests to monitor any potential off-target effects of the CRISPR system. To wrap up, gene editing, particularly through the use of CRISPR, opens up a world of possibilities in treating genetic disorders. The Brilliance trial has demonstrated that this technology can be safely used to improve vision in patients with inherited blindness. If you found this video insightful, do share it with others who might benefit. And while you're at it, don't forget to hit the like button and subscribe to our channel, Science Sparkles, for more illuminating content on the wonders of science. Until next time, keep questioning, keep exploring, and keep sparking your scientific curiosity.